Hi, my name's Threa Roberts and welcome to Body Talks. Today we're going to be talking about antibody structure. So from high school or college or university, when you're first introduced to antibodies, you're introduced to them as these little Y-shaped molecules, like the letter Y, but they're actually much more complicated than that. So let's take a look. Antibodies are also known as immunoglobulins. Immuno meaning that they're part of our immune system, and globulin because they're globular proteins. So these antibodies bind to foreign substances or pathogens that might enter our system, and they mark them for destruction or removal from our body. Let's take a look at the structure. So they're made up of 4 polypeptide chains. They consist of 2 identical light chains, which you can see in red here in this diagram, and 2 identical heavy chains. So the heavy chains have a high molecular weight, they're depicted in blue here, and the light chains depicted in red have a lower molecular weight. So that's where they get either light or heavy chain referral. So they're held together, these 4 polypeptide chains bound together by these covalent bonds, which we call disulfide bonds, or you might hear them referred to as disulfide bridges. There's 5 different types of heavy chain, which determines the class of antibody. So they can be either IgM, IgD, IgG, IgE, and IgA. We're going to look at that further on in the slide. The Ig part just means immunoglobulin. There's also 2 different types of light chain that can be either kappa or lambda. So each antibody can have only a kappa or a lambda light chain, never both. Here is a more realistic version of an antibody. You can see here that there are globular proteins with individual chains sort of coiling around one another. You can also see a small carbohydrate unit between the 2 heavy chains depicted in a sort of indigo colour towards the bottom, and this helps sort of stabilise the antibody structure. Just sort of between the middle of the antibody molecule, we have a hinge region. This hinge region sort of allows flexibility of the antibody and sort of allows it to bend without sort of snapping. So each of the light and heavy chains in an antibody has different regions or domains they're referred to. So they're either a variable domain or a constant domain or region. So we can see the variable and constant regions of a typical IgG molecule in this diagram. The variable region contains the antigen binding site. So this site is found in the N-terminus of the polypeptide chain at the tip of the arms of the Y molecule. The N-terminal half of the heavy chain and all the light chain together make up what we call the Fab fragment or antigen binding fragment. So it has a unique structure which sort of binds to, complementary binds to a sort of specific antigen. So it's the amino acid sequences in the Fab fragment or the Fab region which are different from one antibody to another, and this is why they're referred to as the variable regions. So the constant region is referred to as the Fc region, and this makes up most of the remaining antibody molecules. So you can see the majority of the antibody molecule does contain actual constant regions in both the heavy and the light chains here. And this region, the constant region of the antibody molecule, is responsible for the biological activity of the antibody. So in humans, we've got 5 classes of antibodies which I already mentioned, and we call them classes or isotypes of antibodies. So we have IgG, IgA, IgM, IgD, and IgE. And they're classed according to their sequence of amino acids in the constant region of the antibodies. So IgG and IgD up here are kind of similar. They've got the same amount of variable and constant regions. The The IgG molecule makes up the majority of antibodies in our blood. So they say up to 70%. I've seen some textbooks saying up to 80% of all our antibodies are IgG type. IgG is the only antibody that can cross the placenta, so it provides passive immunity to the fetus. And it's also the major antibody found in the secondary immune response of our immune systems. IgA it can be found as a monomer, just like the Y-shaped molecule, or it can be found bound to another IgA molecule, which we call a dimer. So you can find IgA in the mucosal surfaces and secretions of our body, such as our respiratory, urinary, and digestive tracts. And it's also the major immunoglobulin found in our external secretions, like milk and saliva and things. IgM is really interesting because it can exist as a monomer, but also what we call a pentamer bound together. So it's found as a receptor on B lymphocyte cells, and it's the first antibody produced in our immune response. So when we have our immune response, you'll have heard of primary and secondary reactions. IgM is the most abundant antibody in the primary response, and IgG is the most abundant antibody in the secondary. Similar to IgM is IgD. It can't form a pentamer, but it's also found on B lymphocytes and acts as a B cell receptor. And the last one is IgE. So IgE plays a significant role in our inflammatory response and any allergic reactions, as well as providing protection against parasitical infections. So as you can see, all of these 5 different classes of antibodies, they all exist as monomers, but only IgA can dimerize, and only IgM can form a pentamer molecule capable of providing up to 10 antigen binding sites. So I hope that information was helpful. Please look out for any future videos on the immune system, which I will be uploading in the near future. So if you liked that video, please like and subscribe to my channel so that you can get notifications for any future videos.